Yes, okay, perfect. Well, it's a real pleasure to introduce Becca back. She's a fifth, fifth year graduate student in my lab. Becca's project is a cool intersection of technology and biology. I'll just give sort of a one sentence introduction. Uh, you know, our microbiomes contain thousands of bacterial species, and we have tens of thousands of phages that interact with the microbiome. And Becca is really sort of pushing the technology forward to figure out which bacteria interacts with which phage. And this is a huge unknown in the field. And we really cannot do this without having technologies to compartmentalize these interactions in droplets. And so that's the technology angle. And the big basic science question is to ask who interacts with who. And I think what's really compelling for us is we're not a droplet microfluidics lab, and so Becca has really been the tour de force in bringing these technologies to my lab and to sort of advance them to address this really fundamental microbiology question. I've just been sort of a silent spectator watching her really build these really cool and complex devices. It's gone through multiple iterations, and it's really a testament to her perseverance and her tenacity to have sort of really made a pretty big dent in this problem. Let's see if I may, she's also had to go through some personal struggles to get to this point, and again speaks to her character as a strong person to come this far. So I'm looking forward to the exciting presentation, and the stage is yours. Thank you.
Okay. Hi, everyone. I'm Becca, and as Watson gave me that great introduction, I'm a fifth year in this program, and I'll be discussing with you my research on how I developed this advanced droplet microfluidics method to essentially characterize phage-host interactions. So overall, I'm going to take you through essentially the iterations that I've been going through for the past 5-ish years of my PhD. So I'll go over a brief background on what exactly we study, which are the phages, as well as introducing the methodology or the microfluidics, as Watson put it. And then I get kind of into the details on how my project has kind of evolved since we originally planned it, as well as some exciting preliminary results. And then I'll just recap with what we discussed so far and some next steps and future directions that we want to take this. So phage-host interactions are ubiquitous and highly abundant. All microbiomes are teeming with phages. And how I describe phage-host interactions is essentially the dynamics between a bacteriophage and its bacterial host or host. And there's various kinds of phage-host interactions, but I'm mainly going to focus on lytic phage-host interactions. So lytic phage-host interaction is when a phage finds its corresponding host, attaches itself to it, and then injects its genetic material inside, and it essentially hijacks the host system so it could replicate various progeny of phages inside. And at some point, the cell will just burst, and then the phages will exit and then go on to infecting other hosts. So they are extremely abundant. So here are just a couple of stats. There's no real way to estimate how many phages and bacteria are in the world, but this is just a small estimate. So in the ocean microbiome, very salty conditions, there are shockingly 5 times 10 to the 12th phages per square meter. So if you can imagine a meter and then box it in, that's essentially how many phages you could fit inside that. There are phages in the soil microbiome too. So there's a lot per gram of soil. And then in the gut microbiome, our microbiome is also just brimming with different kinds of pathogens, as well as different kinds of lysogenic or temperate phages that kind of maintain our microbiome. So we're specifically interested in the gut microbiome, and there are so many different kinds of bacterial species with them, and then even more different kinds of phages that target these bacteria. So the relevance of phages is actually quite broad. So here is a slightly busy significance slide, but it just shows how much phage-host interactions kind of impact our everyday life, kind of research the biology as well. So there's a lot of interest in studying bacterial evolution and resistance. So since antibiotic resistance is a very severe occurrence that's going on, we are interested in using engineered phages to kind of counteract these infections. And there's also a lot of interest in kind of engineering these phages. So our lab is really interested in recombinering or making deep mutational libraries and engineering these phages so we could target them for various different applications, such as, for example, targeting different starter cultures in cheese or milk or products for shelf stability, or reducing foodborne illnesses in livestock, such as salmonella. So there are various directions that we could take phage research. So these interactions are highly species specific and also really nuanced. So phages are very specific towards certain kind of strains of hosts or species. So they'll directly be able to infect, for example, indicated by these arrows, this kind of bacteria. And maybe there's like some condition that enables them to target this kind of bacteria with a certain kind of phenotypic or genotypic component. But that won't be the case for all of them. So it's extremely detail oriented and kind of hard to parse apart that. And it gets even more convoluted, as you can see, if we compare mixed microbial communities. So this could be microbiomes where there are various different kinds of bacteria, various different kind of phages. So it's really difficult to really understand who is interacting with whom, which phage is interacting with which bacteria, and also in the context of different microbiomes, we also have things like quorum sensing or nutrient cycling that's going on with other bacteria too. So there's a lot of moving parts in phage-host interactions. And there are various methods to kind of study these different kind of interactions. All of them have their benefits and some of them have their limitations as well. And how I kind of perceived it is in a way of measuring it through throughput, which is low throughput or high throughput. So how many samples you could kind of process at once, or resolution. So how specific can we go? Is it going to be pretty detail oriented down to the strains, or is it going to be pretty broad where it's just general taxonomy? So the gold standards that we use commonly are the culture-based methods. So this is where we have a monogeneous culture of different kinds of bacteria and phages and essentially just mix them together and see if we see some kind of phage activity, whether it's lysis. And then we have a little bit more advanced methods such as computational or data-driven methods where you could study different kind of k-mers or different signatures. And while these give a lot of data, it still needs to be empirically tested. So this is where my research kind of comes in, where we could kind of mix these predictions and actually test them in a way that saves a lot of time. So this kind of highlights a high throughput and high resolution research opportunity where we want to develop a tool that could kind of demystify or kind of help us understand a little bit better about how we could study the individual pairwise interactions between phages and bacteria in really complex communities. So what our goals for this is essentially building a technology that can reflect the natural phage biology. So that would be the lytic phage infections, but also being able to individualize or compartmentalize these interactions so we're able to identify exactly which phage is interacting with which bacteria. Another thing is, most importantly, in my aspect of my project is it high throughput? Is it massively parallel? Are we going to get lots of data that we could actually test and be able to empirically validate as well? And then an additional layer is, can we go a little bit more deeper into that? Can we find the little details such as different kinds of genotypic or phenotypic heterogeneity? And then also, can we make this tool generalizable? Is it something that can only be used in our lab or can it be used in other aspects of research as well? So that's kind of our goal with this project. So this is where the microfluidics come in. So microfluidics is the study of fluids at the sub-nanoliter scale. So this is where you can essentially do stuff such as making droplets, or you could culture things in very small reactions. And the advantage of this is that you could generate millions and millions of reactions within minutes, and it doesn't take that much time, that much effort. It's also highly customizable. So as Watson mentioned, we could essentially make different kind of microfluidic devices that could be customized for different kinds of workflows. So here are just some examples of some devices that I have fabricated in our lab. And this could really go scale between really simple microfluidics that we all know, which are essentially just making droplets, so stochastically capturing cells in very small volumes. And this is where systems like the 10x system is common for this. But then we could also scale towards more advanced microfluidics where you could do more complex liquid handling. So these are just some videos. So these are just some droplets being generated right here. In this channel right here is oil. So since oil is a higher density, it'll basically be able to encapsulate this aqueous layer here. So you'll have essentially an emulsion of tiny, tiny droplets. This right here is a pico injector. So you could see that some droplets are coming down. And if you want to inject a very, very, very small volume of a certain reagent, then you're able to do that with this. So we kind of wanted to test whether we can use a simple droplet maker to essentially do single cell sequencing. So single cell sequencing has been around for over a decade now. There's RNA-seq, there's ATAC-seq, whole genome sequencing in droplets. And what we really wanted to do is kind of take that and advance it towards the microbiology field or the phage field in general. So what we really want to do is ensure that we're creating a technology that is still replicable and comparable between bulk conditions as our gold standards are, and then also be able to reflect that inside droplets. We also want to ensure that we could actually identify these unique phage-host interactions that we might miss in bulk settings, and also be able to quantify. Since we're studying lytic phage host infections, we really want to see whether there was a success in the number of progeny that was produced. So here, this is based on a method from Lan et al. That was published last year, where essentially you just encapsulate bacteria in a PCR mix. And this is a one-pot, very easy, straightforward method where you just encapsulate whatever target template that you're doing inside a PCR mixture. And then you essentially just do PCR amplification. And for this, I kind of tweaked it so I could actually do the phage host interaction studying. So I kind of modified it in a way where I'm still having the phages and the bacteria in a PCR reaction mixture, but then also incubating it because that's how you're able to have the phages be able to infect and replicate. And then after that, I just essentially just PCR amplify it. So this is essentially how I'm able to identify the contents of each cell. It uses a barcode schemata. So we are using random NNN primers that are loaded at limited dilutions. So approximately 1 in 10 droplets will contain a barcode, sometimes even less. And then what the barcode does is essentially having an overlap sequence as well as a P7 at the end for Illumina sequencing. And then you also have a constant region that attaches to whatever target that you want to identify your phage or bacteria. And that attaches to a P5 adapter, so you're able to Illumina sequence. So essentially, it's a two-part PCR or two-step PCR where you're amplifying your barcodes first. And then the second step is where you're amplifying the amplicons as well, and then basically attaching the Illumina sequence adapters at the end. So what we get at the end after sequencing is a bunch of reads, but these reads will have a barcode, and each barcode will be unique towards the contents of each droplet. So no droplet will have the same kind of barcode because that would indicate it's from the same cell. And how we want to quantify this is essentially if there is a successful phage-host interaction, we're going to see phages being able to replicate and lyse the cell. But then when we do the demultiplexing and looking at the data, we'll see that there would be a higher abundance of phage template that is barcoded compared to bacteria. And vice versa, if it's a non-functional phage-host interaction, it will basically be a null reaction where the phages are not able to create progeny, so there is a fairly even amount of template. So we should see really no changes in the number of reads that map towards the phages and the bacteria. So one important question is, how do you know that you're actually getting a single cell suspension? It's not single cell if you can't really show that you're getting individual phages in each droplet. And I do this based on the Poisson loading distribution, which is commonly used in single cell sequencing. So essentially, if you could imagine you have a bunch of buckets or containers and you're throwing a bunch of balls randomly into all these containers. Some of these containers might have 0 balls, some might have 1, some might have 2 or 3. And overall, the Poisson loading or the lambda right here is the mean number of balls per container. So ideally, the lower the Poisson loading is, that means that you have a higher chance of having just one particle, phage, ball, as I said, in each droplet. And then as the lambda gets a little bit higher, that means you have a higher concentration or a higher likelihood of having various particles per droplet. So I demonstrate this by doing some encapsulation of phages, and then I just PCR amplified it and stained it with cyber green. And you could see right here that I have different kind of lambda values increasing from 0.1 to 5. And then right here in the blue is the experimental, which is from my experiments, and the green is what we hypothesized from the models. And you can see that the numbers actually align really, really well if you compare droplets that are positive for fluorescence or if they're negative for fluorescence, as indicated by the 1 and the 0. So this is very promising, showing that we actually can fine-tune the resolution, and we're off to a pretty good start. Another important thing that we kind of wanted to ensure is going on is, are the phages actually infecting? Are we actually getting progeny? And what we saw from our preliminary research is yes, if we compare bulk settings where we're taking milliliters of a certain bacterial host, which is E. coli 10G, as well as our T7 phage, which is E. coli specific, and mix them together, you could see that the titers or the number of viral particles that we get back are pretty similar between each of the conditions. So over here, this is the T7 no host in droplet form, and then this is with the host. And then on this side, it's in the bulk setting. So you can see that the numbers are fairly even, showing that we actually get pretty accurate capture back. And regardless of droplet size, whether we scale from very tiny droplets, which are 20 microns in diameter, to larger droplets, which is 100, and then scaling the concentration appropriately, we could see that the titers are still pretty comparable between these bulk and droplet settings, indicating that we're off to a very good start, and it's actually really comparable to our gold standard methods. However, we did run into some limitations of this simple droplet maker. Um, we When we encapsulated the phages in the PCR mixture for the one pot reaction mixture, we found that the phages were unable to infect the host. We attributed this due to the salt conditions of the PCR mix. And what we tried was trying to add more LB media in order to, kind of supplement the conditions. Hopefully, like the phages will be a little bit more happy with a little bit more LB media. But you could see in these gels right here that there really isn't, these gels and this bar plot right here, that there really is no difference. So over here on the left, this is with the ddPCR environments, and you can see that the titers don't increase at all. Comparably, this is their control, which is an LB media, and you can see that they should have increased. And another hiccup that we kind of ran into was that even if we did want to add more LB media, we found that there was PCR amplification inhibition as well. So this is comparing phage-only samples and phage and host samples with various dilutions of LB. And you could see that as you get to the larger amounts of LB in bulk and in droplet forms, you could see that there's less and less template in these gels. So overall, we are trying to go through these steps to kind of validate this method, and we are finding that we are so close. We were able to get to the single cell encapsulation phase. We theoretically were able to get phage replication, but as for the sequencing, we aren't going anywhere with that. So this called for alternative methods. So this kind of called for a more advanced version. Up here, we had the simple one pod, just encapsulated in PCR, plug and chug kind of method, and we found we can't do that at all. So that means we have to move on towards a little bit more advanced liquid handling methods right here. So what if there was a way where we could separate the biology from the molecular biology and then combine them together at the end? That way that we're kind of allowing each to do their thing without inhibiting each other. So there are various microfluidic methods to basically combine or mix reagents together. So these are just two examples, one called pico injection and one called merging. Pico injection is essentially where a small volume of a reagent is injected into droplets right here, versus in merging, you have two droplets that are coming together and being able to destabilize and then couple back together. So these are just some that have emerged in the past decade or so and is commonly used. And there's various different kinds of designs, all claiming to be very efficient. And if you're in the field of microfluidics, you know that it's not really that. You have to actually make it yourself and test it to experience this. So as I said, we're going to use this advanced microfluidics method instead. So what we're planning on doing is just encapsulating the phages and bacteria in LB media. They're happy places essentially, so they'll be able to replicate. And then generating PCR droplets on its own that have the barcodes in it, and essentially mixing them together. So as they come towards an electrical source, the surface of these droplets will destabilize, and as they move away, they'll restabilize, resulting in a combined droplet. After that, we'll just PCR amplify it, each attaching their appropriate barcodes in a Illumina sequence, and then demultiplex at the end. So there are various different ways that I had to go about this on how to design this. There's various designs out there, but none of them were really appropriate for my kind of experimental setting. So I had to kind of dig and think about how I'm going to design this device. So there are various different kinds of parameters that I had to consider. Droplet sizes, for example, if there was too much LB in one droplet, that means that we're not going to be able to get PCR replication when we combine them together. And I'm working with electricity, so too much electricity, you're going to have stray voltage that might affect other parts of the microfluidic device. So we didn't want that. So there was a lot of fine tuning going on to ensure that we're able to get a stable device going. And this is where a lot of my PhD kind of happened, which is trying to get an optimal device working, where we could still kind of have it at the single cell resolution where we're having a droplet that has a phage-host interaction and making sure we're not getting duplicates of each or duplicate barcodes essentially coming together. Good news is that we kind of optimize these parameters, and we have some pilot studies that I'll be showing soon. So yeah, this is a familiar figure that I showed before, but it looks complicated, but the three main structures that are the most important for doing anything with droplet combinations or reagent injections is that there's an electrode. So could be saline, this could be liquid metal, this could be as simple as a paper clip in my case. And essentially, this electrode causes a voltage that goes towards a certain site, combination site, and this essentially just destabilizes the surface of whatever liquid's going on. As it moves farther away from the combination site, it restabilizes. There's also a moat that is commonly used to kind of ground the electrical voltage to ensure that it's not going into any other direction. For example, if you're injecting other droplets in another position, you kind of don't want to ruin the integrity of those droplets. So this is my little setup that I have. I have a little function generator to create my volts, and then a voltage amplifier, my little oscilloscope where I could actually visualize different wavelengths, and then my microfluidic setup that I could go into a little bit more detail later on. And what we should be able to see is that the voltage should be able to merge the droplets. So you could see droplets being generated from the left here and then some on the right. This is with no voltage, and you can see that they do not combine together as you see them towards the end. Here is with voltage, but you could see that the droplets are different sizes, which means that there's too much electrical signal going on, which is causing the droplets to destabilize at some point. So there's a lot of fine tuning that had to go along with that. In addition to that, I had to think about how I'm going to design these combination sites essentially in a way where we're able to get electrical signal that is very specific towards certain sites, but also making sure that we're not giving it too much electrical signal where it's being brought elsewhere. So I have two examples right here. In this example right here, there was basically only one way in for the electrode to go in, and all these little clumps that you see here is salt. So it was literally cooking the salt with the voltage right here. So this design was not viable. I had to change it every so often with the experiment. So that was not ideal. And then here's another example where I tried to have various outlets where I could flush out the saline a little bit, but there are air bubbles in this, which means that the electrical signal is not able to reach the merging sites. So these were also some things I had to take into consideration. So this is where I came up with the Kirby merger. So essentially, I have this droplet device where I have a simple droplet maker up on the top, and then I have our merging chamber right here, or the combination site, as well as the grounds to make sure that the electrical signal isn't going anywhere, as well as the electrodes right here for the electrical signal. And then over here, I call it the Kirby merger because Kirby's a little character that is pretty adaptable, resilient, needs food and sleep. And I thought, what's a better mascot for a grad student than Kirby? So the saline reservoir, or the Kirby, is kind of holding some of the saline. So in case there's any hiccups with the electrodes, I'm able to flush more and more without any issues. So these are some experiments where I had to kind of tune the droplet sizes. If the droplet sizes are too similar, then you're actually going to get a lot of droplets pairing together, which is not ideal. You want a one-to-one pairing ratio. And then I was able to kind of get to that by having a larger droplet with the PCR reagents and a smaller droplet with the phage host interaction. So you could see the droplets are coming from here, and then they're merging together as they come towards this electrode site. And you're probably wondering, how do you know how efficient your merger is? Well, what I did was I took droplets that had yellow dye in it or blue dye in it, and I took images of before and after merging, and then essentially did image J analysis where I measured the diameters and I quantified all of these droplets. And you could see that I have a good percentage of overlap right here on a shift in diameter, indicating that we have a lot of droplets that are merging together. So in this experiment, we have about 70% of the droplets actually merging together. So that's a 70% merging efficiency. And I kind of validate this a little bit more by doing some simulations. So there are some computational fluid dynamics simulations that I can use to further validate that this microfluidic device is actually working. So a benefit of this is that we actually see the 3D fluid dynamics versus the point-only data we see in the imaging. And then this also helps because it gives me an idea of what parameters I should tweak in the future. So this is one example of what I can do with computational fluid dynamics. This is velocity along the path. And essentially this predicts whether there's any kind of turbulence or any kind of parameters that might mess up the experiments. So you could see each of these colors are highlighting a certain region in the microfluidic device. And as I would anticipate, there is a little bit more, you could see shakier or little waves going on for the velocity going on in the merging chamber. And that's what we are expecting because that structure is a little bit more dynamic compared to the rest. Over here is another image of the droplet generation site. You can see that there's a little bit more velocity and pressure going on here, and that's because there's a higher flow rate going towards making the droplets. And over here, you could see the same. Since this is a tight space, you could see that it kind of constricts and then it releases to a higher pressure as it moves further away from this constriction site. And this was fun, the merging chambers, especially because we wanted to make sure that the fluid dynamics actually occurs with a one-to-one droplet ratio. And you could see that the velocity goes along the walls of the channels right here. The pressure is pretty consistent and high towards these square channels right here, and it kind of loosens up as it goes towards these squid game channels, as I call it. And that involves the droplets being able to slow down, come in contact with each other, and then merge. You're probably wondering, will this PCR amplify? And the good news is, yes, we are able to get PCR amplification after merging these two droplets together. So over here, I have a non-template control merged droplet on the left. You can see on this tape station right here, there's no peak or no amplification versus a droplet that has actual phage template in it. You could see that it was able to create a pretty clean amplicon. So I did a pilot experiment where I essentially took T7 phage and K1F. Both are E. coli phages, but they're dissimilar in the way that K1F can only target biofilm creating E. coli. And I chose a region to target that is dissimilar enough where I'm able to distinguish between these two samples. And if you look at this barnyard mixing plot right here after sequencing, you could see that I'm not getting any kind of duplicates, very little duplicates of T7 phage and K1F, indicating that I'm not getting not as many two cells per droplet going on here. And you can see that I have a fairly equal capture of both phages as well. So I kind of want to conclude with the RCR portion. So I chose to focus on ability or disability as well. There's nothing wrong with saying disability. So there are civil rights laws that kind of prohibit disability discrimination, help provide access and reasonable accommodations, and to protect individuals with disabilities. And especially in academia, there's a very small margin of people with disabilities, even though there are a bunch of different civil rights acts that have been established to protect us. But there are ways that we could still be included in research settings. So some examples of some provided accommodations that occur in our lab is very clear and concise communication. So this is to kind of straightforward, what do we have to do? This reduces any kind of cognitive load or fatigue or trying to read, each other's minds. We try our best to communicate with each other. We also try to make more flexible schedules. So this could be either if you're not having a great day body-wise or neurologically wise, maybe you could come in later, or maybe you could work at home. So there's that you can also do, as well as task redistribution. So my lab has been so great in lab chores. For example, when I hurt my shoulder, I wasn't able to do a lot of lab chores, carrying dishes and cleaning things, so they helped take over. And then as well as safety and environmental, we came up with a seizure first aid safety plan, and I essentially introduced everyone to this training. So everyone in my lab is seizure first aid certified. So, yay. So yeah, in conclusion, I kind of went over my whole entire journey of how I kind of went from this very, very ambitious project that evolved into a more advanced kind of technology. And we're really excited to see where it goes in the future where we actually test these phage-host interactions. And that's it. Yeah, I went a little bit over, but yes.
I'm curious, do you think that you'll get resolution enough in the sequencing abundance to be able to tell like the, I guess, extent of infection? Like, could you be able to distinguish a weak phage host interaction where maybe there's only a few progeny created to like a really strong one?
Mm-hmm.
They're gonna be more kind of binary.
We are expecting it to be a little bit more binary, but droplet PCR reactions are pretty sensitive, so they amplify a lot more differently compared to bulk settings. So we do anticipate like a slight uptick in changes between quantities of different targets. So that's one advantage of using droplet microfluidics versus in bulk settings too, because we might not be able to see those discrete changes.